Hello everybody, it's Kenneth, and today I'm back with another video. And in today's video, we're going to be talking about nano devices, particularly with respect to medicine, of course. And the slideshow is not mine, so if you want to see whose it is, feel free to pause and read. But let's get into it. So, what is nanotechnology? Well, a nanometer is a billionth of a meter, which is really small. Or you can think of it as like 1/8 out of 80,000th of the width of a human hair. It's very small. Okay, and these can be used to create useful materials and devices in such a small scale. And this can be helpful for many disciplines, not just medicine, but today we're just going to be talking about the medicine aspect. So, a lot of the research today, or rather in the past when the slideshow was made, was designed to reach a better understanding of how matter behaves on the nanotechnology, like nanometer scale. And the factors that govern like systems on our scale, larger scales, don't really apply there necessarily because nanomaterials have very large surface areas compared to volumes. Things like friction and sticking are relatively more important there than to larger systems. And if you're into biology, you'll understand that the surface area to volume ratio is a very important concept for biology. Yeah. So there are some challenges that apply to using nanostructures within the body because they can be so small that the body may clear them too fast to be effective, or larger ones may accumulate in organs, creating an issue like a toxic problem. So basically, researchers and scientists have to consider these when creating the devices. Now, there are two basic approaches for creating nanodevices. They're the top-down and bottom-up approach. The top-down approach involves molding or etching materials into smaller components and has traditionally been used in making parts for computers or electronics if you want to look up videos for those. And the bottom up approach involves assembling these structures atom by atom or molecule by molecule and may be more useful for manufacturing medical nanodevices. So, most animal cells are 10,000 to 20,000 nanometers. So, nanoscale devices, which are typically less than 100 nanometers, can enter these cells and the organelles inside them to interact with DNA and protein. This is very helpful because it allows cell specific interactions. And tools developed through nanotechnology can then be able to detect disease in specific cells instead of an area. And that will be helpful, and we'll see specific examples of the nanodevices soon. So, detection of cancer at early stages we know is critical, as we talk about in imaging in other video. And current detection usually depends on changes in cells and tissues that are detected by like touch or imaging, right? But with nanodevices, scientists could detect molecular changes as soon as they sprout up, and that will help reduce the mortality of cancers. So, in order to successfully detect cancers at its early stages, the smallest molecular changes have to be able to be detected, right, with these devices. So, the tools have to be very sensitive, and nano devices can meet these needs. Now, many of these tools could make it possible for doctors or clinicians to run tests without actually altering or removing cells from a patient. And that's important because uh, like screens for cancer are often in limited supply, like the samples for them. And it's important because it can capture and preserve cells in their active state. Like when you remove cells from the body, they're no longer alive, they're no longer active. But by sending devices into the body, they can stay active, and they do stay active. So nano devices can also make it like easier for cancer tests, and miniaturizing these tools can allow many to be on the same device, which can allow many tests to be run at the same time. Now, one nanodevice that can improve cancer and diagnosis, detection and diagnosis, is the cantilever. Now, these levers are anchored at one end and can be engineered to bind to a specific molecule that represents a change that comes with cancer. And then they can bind to altered DNA sequences or proteins that are present in certain types of cancer as well. And when these molecules bind to the cantilevers, surface tension changes, which causes the cantilevers to bend. So if we can monitor which ones bend, then we can tell whether the cancerous molecules are present, which can prove effective in finding cancer within the body. Now, another interesting device is the nanopore. Now, this is a method for reading the genetic code within a person's body and can help detect errors in genes. Now, nanopores are tiny holes that can allow DNA to pass through one strand at a time, and this can make the sequencing more efficient because as DNA passes through a nanopore, scientists can monitor the shape and electrical properties of each base and therefore determine what your gene sequence is. And because these are unique for the 4 bases, all the properties of the bases are unique, then the scientists can decipher the information and find errors that may be helpful. Now, another nanodevice that can help identify DNA changes is the nanotube. Nanotubes are like carbon rods, and they're half the diameter of a molecule of DNA, so very small. And they don't only detect the presence of altered genes, but they can help pinpoint the exact location of the changes. Now, what's interesting is that to prepare for DNA for this analysis, scientists must attach a bulky molecule, like just a larger molecule, to regions of the DNA that are associated with cancer. And to do this, they can design tags that seek out those specific mutations and bind to them. So once a mutation has been tagged by the bulky molecule, a nanotube tip resembling a needle, such as a needle on like a record player or something, can trace the actual shape of the DNA and then further pinpoint the mutated regions. And this basically what it does, okay, is it creates a map of the DNA molecule and identifies mutations. That's basically what it does, and so that's like really all one has to know. Now, quantum dots are like tiny crystals, and they glow when they are stimulated with UV light. So the wavelength or color of the light depends on the size of the crystal. Now, latex beads filled with these crystals can be designed to bind to specific DNA sequences. And then by combining the different quantum dots within a single bead, scientists can create like probes that release distinct colors and intensities of light. And when the crystals are stimulated by UV lights, each bead emits light that serves as a sort of barcode. And so it can identify a region of DNA. So basically what these do is they identify, once again, regions of DNA that may be mutated. Now, quantum dots can find cancer signatures because they can bind to find specific barcodes of DNA. And so when they're stimulated with light, they emit their barcodes, their light waves, and they show the DNA sequences that are associated with cancer. And so, since there are so many types of quantum dots, sizes, light waves that are released, many labels can be created to identify many regions of DNA at the same time. And this is important because cancer comes from many different changes within a cell. So, it can also be useful for developing ways to kill cancer cells or treat cancer cells. And so scientists hope that nanotechnology can do this, and it's promising that it can. For example, these nanoshells, they're beads that are coated with gold, and they can absorb specific wavelengths of light. Now, the most useful ones absorb near infrared light, but basically what it does is once they absorb the light, it creates an intense heat that is lethal to kill cells. So imagine that these nanoshells, they attach to a cancer cell or go inside a cancer cell, then the light wave is introduced to the cells or the body, and that causes the nanoshell to heat up, and then the cancer cells die. So as long as nanoshells can be linked to antibodies that recognize cancer cells, and they can, then they can be sent into the body to kill specifically cancer cells. So a great treatment that has many less side effects than like traditional treatments. Now, researchers aim to eventually create nanodevices that do much more than just deliver treatment. They would like to create like a nanodevice that can do everything, like maybe recognize a tissue or recognize that a cell is cancerous, report that it's cancerous somehow, and then also like kill it. So like everything in one. And so one of these could be like dendrimers, but dendrimers are also nanoparticles that can facilitate drug delivery. Now, they are about the size of like a protein, and the shape gives a lot of surface area, so many like therapeutic agents or drugs and other molecules can be created to attach to the dendrimers and therefore do many things within the cell. Now, a single dendrimer can carry like every type of molecule. As you can see on the screen, it could carry a detector of cancer, a reporter of cancer, a treatment for cancer, and something that tells us whether or not the cell is dead. Now, right now, I don't know how popular exactly nanodevices are or if they're even in the field yet in practical application, but when they will be, they will definitely be useful. That's really all I have for you guys today. Thank you guys so much for watching, and I hope you enjoyed.